Okay, looks like that's happening. Let me just make sure. Okay, great. And here is our lineup. We have four amazing speakers, Sandy, Cathy, Betty, and Tian, and two wonderful moderators, Stacey and Lewis, who are going to be guiding us through this webinar. And with that said, I am going to stop sharing my screen, and I'm going to invite Stacey and Lewis to come to the proverbial stage and get us rolling here. Thank you all so much.
Thanks, Tim. So I'll start off and just to say it's a pleasure as always to welcome everyone to an HVAC conversation. We've got really a great group on the line, so excited to have a discussion with you all and really dig into this topic of discovery or experimental medicine. You know, every year we really try to convene a kind of a timely discussion about vaccines, and I think folks are aware that over the past several years, vaccine research has really kind of shifted upstream, kind of away from the more classic product development approach and into what's sort of being called experimental or discovery medicine. And we're going to hear all about this from our speakers. So I'm not going to go into any detail on what that all means. But for those of you paying kind of closer attention, you know that AVAC did actually focus on this, I believe, for HVAD, a couple of years ago when this shift was first starting to happen. But we felt like now was sort of a good time to circle back and sort of take stock of where we are and what it means for vaccine research and for prevention broadly. Everyone knows that right now the prevention landscape is really evolving, and we've got improving new and improved PrEP products. And there's even, talk about being able to end the epidemic with what we currently have. So at AVAC, we're really, really focused on maintaining kind of a sense of strong urgency around R&D as critical to ending the epidemic, even this very early kind of upstream R&D. And so for vaccines, especially with this sort of shift to kind of this experimental or discovery medicine approach, it's really more important than ever to be able to kind of tell the story of how we know that this approach is really kind of the most promising way to get us to that ultimate end goal that we all know we need of developing an HIV vaccine. So we've got a really great list of speakers, as Jim said. But first, I'm going to pass the mic over to our co-moderator, Lewis Shackelford from the HVTN. And Lewis, just to say, we know the HVTN plays just such a critical role in vaccines, as everyone knows, in discovery medicine. And we're really honored to have you on here to co-moderate this, and just thankful for your partnership and the partnership of the HVTN.
Thank you so much, Stacey. It's an honor to be here, and thank you to AVAC and the Choice Agenda for having me. It's really an awesome privilege to share space with you all. So for those who may not know, my name is Lewis Shackelford, him pronouns. Sorry, it's early on the West Coast here in the United States. But my role at the HIV Vaccine Trials Network is the Director of External Relations. So in my role, I manage our stakeholder relationships, and I also manage some of our awareness and educational initiatives, such as the HVTN Faith Initiative. And so just wanted to get us started off on HIV Vaccine Awareness Day. Why is it important? Why are we here today? So it's really important to remember that HIV Vaccine Awareness Day is also known as World AIDS Vaccine Day, and is celebrated annually on May 18th. It was first observed on May 18th, 1998, and this day commemorates then President Bill Clinton's pivotal 1997 commencement speech at Morgan State University. Morgan State is an HBCU, or historically Black college, in Baltimore, Maryland. And at this commencement speech, President Clinton stated, "Only a truly effective preventative HIV vaccine can limit and eventually eliminate the threat of AIDS." This speech helped to build momentum for some of the initiatives represented on our webinar today, such as the HIV Vaccine Trials Network. And we are the largest publicly funded clinical trials program devoted to the development of a safe and effective HIV vaccine, and also initiatives like AVAC. So Clinton's declaration also led to the development of the the Vaccine Research Center, which has become an important hub for vaccine research across diseases such as HIV, COVID-19, and others over the years. So we mark this day by promoting the continued urgent need for a vaccine to prevent new HIV diagnoses and HIV-related deaths globally. We also acknowledge and thank the thousands of volunteers, community members, health professionals, advocates, supporters, and scientists working together to find a safe and effective HIV vaccine, and urge the international community to recognize the importance of investing in new technologies as a critical element of a comprehensive response to the HIV/AIDS epidemic. And so with that, I want to get us started on our presentations today. And so first, I want to introduce my colleague, Dr. Sandhya Vasan, Vice President of the Henry Jackson Foundation, Global Infectious Diseases, and Director of the HJF component of the U.S. Military HIV Research Program. So with that, I'll hand it off to you, Sandy.
Thank you, Lewis. Can everyone hear me?
Yes, loud and clear.
Yes, we can. Yep.
Okay. And give me 1 second to ensure that I'm sharing the right screen. Can you see my slides in full view? Oh, not yet. Hold on. Can you see that?
They look good, Sandy.
Okay. Well, thank you everyone for joining from all over the globe, especially those who are joining very early or very late. And most importantly, thanks to AVAC and the Choice Agenda for organizing this and giving me the honor of kicking off this important webinar in honor of HIV Vaccine Awareness Day, or World HIV Vaccine Day. As Lewis said, I'm Sandhya Vasan, and I'm with the Henry Jackson Foundation, and I help direct that component of the U.S. Military HIV Research Program, which is another large publicly funded network of global trials to advance the development of HIV vaccines and therapeutics. And we're all one family. We work very closely with the HVTN and others to advance our collective agenda. And so what I was asked to do today was to talk specifically about experimental medicine or discovery medicine with a lens towards HIV vaccine development. And so to do that, I will just start by giving a background of clinical trial development in general to helps to set the stage to explain how experimental or discovery medicine might be slightly different, a variation on that theme, and then end with a couple of examples of these types of trials. And I'll try to go fairly quickly so that I can give time to our other speakers, because I really want to hear them, but also look forward to discussing questions afterwards in the Q&A. So just to start with the basics, how are vaccines developed? Generally, they're developed by screening and testing different potential vaccines first in the laboratory, looking at some of their qualities or their ability to bind antibodies, other things that we can do just in a laboratory setting. And those that look promising, we take into animal models. And those from there that look further promising, we then move into clinical trials in humans. And from there, those that are the most successful get licensed by, in the United States, it's the Food and Drug Administration, but around the world, there are many parallel organizations that provide approval that these vaccines have adequately been tested to be safe and effective to move for widespread utilization in people in that country. And so these stages tend to progress in a linear fashion in the more traditional product development model, where they'd start in the animal models and then move into what's called a phase 1 clinical trial, which is usually a small number of people that simply tests for the first time putting in people, are these products safe? And then if they look safe, although we can also measure immune responses in those trials, they will progress to a phase 2 clinical trial, which is the same sort of thing, but in larger numbers to make sure that there aren't any rare side effects that might be missed in the first smaller trial. And if those trials look promising, they then move into what are called phase 3 clinical trials, which are much, much larger and assess whether the vaccines actually work in preventing the disease that they're meant to prevent. And if they look like they have been successful, then they can move to licensure. And so how do those phase 3 trials work? I'll outline on the next slide. These are called efficacy trials because if a vaccine works at preventing disease, it's considered effective. So the way to test this is to enroll a large number of people, as I said, specific to HIV. These people, if these are HIV vaccines, then these people should be have some risk of maybe acquiring HIV. And then they are randomized, so neither the study team nor the participants themselves will know if they're getting the vaccine or what's called a placebo, which is, something that looks like the vaccine but is inactive. Oftentimes that's just as simple as saline or salt water. And then over time, and then all of those participants are counseled, of course, on how to keep themselves safe and to avoid HIV infection. But then over time, both groups are counted to see whether the incidence or the rate of people acquiring HIV and those who got the vaccine or those who got the placebo, to see if there was any difference in those rates between the two groups. So I'm going to give you one example of a study like this that I was involved in the follow-up to this trial in Thailand called RV144, or more generally known as the Thai trial. And just to give you a little flavor of how this is actually conducted, this particular trial enrolled men and women who were ages 18 to 30, who were at what was considered community risk. And it really required enrolling a large number of people just to get the outcome to be able to assess whether the vaccine had any efficacy. So over 16,000 people were randomized, roughly half and half, to get vaccine or placebo. I won't go into the actual vaccine regimen, but it's depicted below, and it was a combination of 2 vaccines given in various combinations over 6 months. And then people were followed for an additional 3 years, or 42 months roughly, and they were enrolled from 2 provinces in southern Thailand. And so this graph shows how we analyze results from an efficacy trial. And so over, across are the number of years that the trial was conducted. And then on the vertical or y-axis are the number of people who actually then acquired HIV over the course of time. And so those numbers, the actual numbers at the very bottom. So at 1 year in the people, the large, out of the over 8,000 people who received a placebo, 30 of those people went on to acquire HIV. And of the 8,000 people who received the vaccine, 12 of them went on to acquire HIV. So then you look at those rates over time and see whether the vaccine looks like it's lowering the rate of people who are then being diagnosed as having acquired HIV. And so at the end of that 3.5 year period, there was a difference between those two groups. The difference was small, so the vaccine was considered to be 31.2% effective in preventing HIV. So on one hand, that was hugely encouraging to see that this could be possible. On the other hand, that difference wasn't enough to actually go ahead and license it. By way of comparison, the COVID vaccines that were tested in their efficacy trials, the first few were above 90% effective. So, it was getting us in the right direction, but we weren't quite there yet. And so building on that, a large number of groups, many of whom are represented on this call, came together in what was called the P5, or the Pox Protein Public-Private Partnership, which is a mouthful. to try and build on that result and move these results into Africa. And so this was conducted through a series of trials, and the major stakeholders from Bill and Melinda Gates, the Division of AIDS at NIAID, U.S. National Institutes of Health, pharmaceutical companies like GSK, Novartis, and Sanofi Pasteur, the Republic of South Africa Medical Research Council, and where I work, the U.S. Military HIV Research Program, were all involved in this massive effort to see, could we build on those exciting early results in Thailand and move them into Africa? First through HVTN 097, which was testing the very same products in South Africa for safety and immune responses, then testing related products that were customized for that region in South Africa, which was called HVTN 100, and then eventually moving to that big phase 3 efficacy trial called HVTN 702 of the products that were tested in HVTN 100. And unfortunately, in February of 2020, it was shown that there was no efficacy, meaning that the rates of HIV acquisition in people who received vaccine or placebo were very, very similar. But what I want to show you here, as you look at that date, February 2020, and AVAC has put together a very nice summary slide about HIV vaccine and antibody efficacy trials to date. So this includes both testing of vaccines and monoclonal antibodies, which are, I won't go into the subject of that in this short time, but another approach that the field is taking to try and prevent HIV. But what I really want to show you on this slide is not the individual trials, but rather just the top row. If you look at the number of trials that are efficacy trials, these large, complex, expensive trials that really involve a lot of stakeholders, a lot of community involvement, they're hard to conduct. And so there haven't been that many of them. And you can see they haven't been that frequent either. So two studies, the one I just mentioned, the RV144 or the Thai trial in 2009, and then a monoclonal antibody study in 2021 have shown some efficacy. And, in light of the fact that this is HIV Vaccine Awareness Week, I don't want to end on a depressing note. We have a number of concepts to build on these to try and keep moving this field forward. And we still remain excited and committed to developing a vaccine to prevent HIV. But what we really need to do, and the focus of this talk, is to see how we can make that move a lot faster. And that's now all just by way of background to explain what we're talking about with these experimental medicine trials. And so historically, through the traditional approach, you can see another nice slide from AVAC that they put together, how long it's taken to develop licensed vaccines through a traditional pathway. But the world came together in the face of the COVID pandemic to develop vaccines to prevent or to decrease the severity of SARS-CoV-2 infection. And we did that much, much more quickly. And it was a wake-up call to realize with the right mobilization and the right resources and the right focus, we can go even faster. I myself have been involved in HIV vaccine development for over 20 years, and I can speak for a large number of us in the field that COVID made us a lot more impatient. We need to go faster. So how do we do that? Well, that's moving to the next part of the talk and what is experimental medicine. And I'll refer you to a very nice paper and a concept sheet with the web link there put out by AVAC and the International AIDS Society and the HIV Vaccine Trials Network that outlines this in a lot more detail. And I'll also take a moment here. Stacey asked me to talk about nomenclature a little bit because experimental medicine is one name for this approach. It's also sometimes referred to as discovery medicine. Sometimes people call it phase 0 trials. They're roughly equivalent and really embody the same concept. Actually, at MHRP, where I work, we don't really focus on this nomenclature, but we're all embracing the same concept regardless of what it's called. And so I I won't walk through every line of this slide. I think it's both on the AVAC website and in the paper. But generally, I'll tell you that the concept of discovery or experimental medicine is not to develop a product in that linear fashion, but rather to test a scientific hypothesis that may more quickly accelerate the next generation of vaccines. And so, that is also outlined in the first line of the table here. But generally speaking, the numbers of these trials could be even smaller than a phase 1. They may require specialized assays or specialized tissue collection. And, it's important to work with the volunteers to understand that there's not necessarily any direct benefit to them, but they are part of the scientific process to provide benefit to the field so we can all continue to move forward as quickly as we possibly can towards our shared goal of developing a vaccine to prevent HIV. And so some of the benefits of these types of experimental trials, as I already said, are that they're smaller and therefore can move more quickly. You can test a concept very quickly without going all the way to phase 3 efficacy and saying, should we iterate this and come back to the drawing board to make it better before we go on or not? Ultimately, we unfortunately still have to utilize animal models to test some of these concepts, but humans are the most reliable way to look at what an immune response will really look like. So we want to be able to do more human trials quickly. But when we go more quickly, that doesn't mean we go any less safely. We maintain the same standards of ethical and regulatory review in all of these trials. And I think that's really, really important to emphasize. We saw some backlash after the touting of how quickly the field went to develop vaccines for COVID that started to create worries that going more quickly meant perhaps going less safely. And that's not at all the case. Every single one of us who works in these types of trials, I assure you, is committed to maintaining the utmost standards of safety and ethics in all of these trials, no matter what they're called and no matter how big or small they are. That said, because of that, what it doesn't speed up is the manufacturing process. Even if you want to test a new concept of a new vaccine pretty quickly in a small number of people, you still have to have it manufactured and go through all of the same qualifications to ensure that product is safe, whether it's in 10 people or 1,000 people. And that takes time. So it's the same thing for the ethical review. The participants really have to understand that they're part of a larger mission with no direct benefit to themselves, and that there may be more extensive involvement of sample collection depending on the protocol. But with that, it's been a successful approach so far by so many networks, the HVTN and others, and I am going to focus on 2 examples from the MHRP here to round out the end of my talk. And the first is a study we call RapidVax, and it's based off of a concept of a groundbreaking study led by Dr. Merlin Robb from the MHRP and HDF with collaboration with many, many, many awesome partners that we have in Africa and Thailand to enroll people prospectively to catch them at the very, very, very earliest stages after their HIV diagnosis. So we could look at what you can see here in the red oval, that look at how the virus comes up in the earliest parts of infection. Those gray lines are HIV viral load on people's blood. And then we can see how in the earliest stages of infection, it goes up, but then it comes down pretty quickly. And then the references at the bottom state a number of animal studies that have also validated this approach, which is depicted in the next slide, and which is a trial that is now enrolling at the Makerere University Walter Reed program in Uganda. Some of the speakers in this webinar are involved in this. And it just shows that relative to a standard vaccine regimen at the top, which is what all the symbols are, and I won't describe all the vaccines themselves, the testing of the idea that mimicking that early viral load upslope and providing vaccines very quickly at increasing doses might make our body see the vaccines the same way we see early virus, and it might help us develop broadly neutralizing antibodies and other immune responses critical to then preventing subsequent HIV infections. And so the point of this is not the science behind it, but rather to show you that that kind of rapid approach, I'm sure many of you would acknowledge, is not feasible for large-scale development. It's really a truly discovery or experimental trial to test this concept. If the concept looks good, then we iterate to say, how can we achieve that in a more sustainable way for global populations? And then the second quick example I'll give you is to end my talk are this concept of multiple founder viruses. And this is out of a paper that's cited here from Dr. Morgan Roland at HCF and the MHRP, who looked at people who acquire HIV. Most people who are diagnosed with HIV acquire just a single strain of HIV, which is called a founder virus. But some people actually have more than one strain or more than one founder virus. And the research in this paper shows in pink those people who started with more than one strain of virus were more likely to develop broadly neutralizing antibodies later in their course. And so how could we maybe take this knowledge and mimic it in a vaccine? Well, the other thing is that HIV strains across the globe, depicted here beautifully and referenced in the paper there, are really, really diverse across the globe. And many of you have probably heard that. But interestingly, in those people who require more than one strain of HIV or multiple founders, that diversity on the same scale as shown there in the black is actually pretty small. So if I'm one of those people who's just acquired HIV with more than one strain, those strains are actually pretty related to each other. They're not highly disparate from different parts of the world. And so we can take this concept depicted in the dots here about the different strains from a single individual and how closely related they are, and optimize those sequences to maximize immune responses, and then put them together in a cocktail. And right now we're actually working to put them together into a single mRNA encapsulation with multiple sequences that are closely related but slightly different. So again, this is another example of this sort of discovery experimental approach, because we don't know necessarily, we don't think that this will be a home run in the first shot, but we're using it to test this novel concept, and we're sharing it with the brilliant consortium that you'll hear about very, very soon in this webinar that's funded by the USAID VISTA program. And with that, I'll just acknowledge the many, many, many groups it takes to conduct this work, both clinical, pharmaceutical, but most importantly are all the study participants and the community support it takes to conduct all of these trials from small to big. And lastly, I'll end with this to say that the community continues to play a key role for the success of these trials, because it requires engagement at multiple levels with the study teams, with the community advisory board, the community itself, and a place where we can use your collective voices also to lobby to help streamline and harmonize regulatory processes so we can continue to get these trials approved more quickly. And thank you for your attention. I'll stop there.
Wonderful. Thank you. Thank you so much, Sandy. We knew you would give a great overview, and you definitely delivered. I do want to take a second to see if there are any burning questions or clarifications from Sandy's presentation. Feel free to jump in and just still continue. I know we've got some questions coming in to the Q&A box, but do want to encourage people as we go to add your questions, because this is a lot of great information. So I'm actually going to turn it over to our next speaker, and we'll have Dr. Cathy Slack. Dr. Cathy Slack leads the HIV and AIDS vaccine ethics group, which is based out of South Africa. And she is going to, really wanted to kind of not get too into the weeds of all the technical science, but also bring in some of the more kind of ethics and community perspectives around this research approach. So Cathy, over to you.
Thanks so much, Stacey. Jim agreed to advance my slides for me. So I'm just going to ask him to do that, please. And just an apology, for some reason, my camera is not being recognized. So unfortunately, I just can't turn on my camera. So my apologies for that. Just while we're waiting.
Oh, I was just going to say thank you, Cathy. I'm going to get your slides up in a second.
Super. Thank you so much, Jim. While Jim is doing that, would just like to make a few remarks about the team. So Siyabonga, Tabeta, Abigail Wilkinson, and I are working on a project that's currently housed at the South African Medical Research Council with Siyiso Kowali as the MRCPI. And we are one of 13 sub-awardees for the CASPER project. And as part of that coalition, what we're really focusing on is how to support the impact of international ethics guidelines. So really, how can we increasingly be part of all those groups that are committed to getting guidelines off the shelf, into the field, into people's minds, etc. Jim, have you got the slide up?
Yes, your slides are up.
Excellent.
Your first slide is up.
Could you please just go to the next slide? Thanks, Jim. So,
sorry, one second, Cathy. I'm not sure if I'm the only one, but Jim, I think we're just seeing a shot of your folder so far. We're not seeing the actual presentation.
Yeah, I think it's Jim's folder.
Okay, sorry about that. Just give me a second.
No problem.
I will restart the screen sharing. Can you see it now?
Yes, that looks much better.
Okay, sorry
Thanks
for
so
that.
much, Jim.
Go ahead, Cathy. Thank you.
It's no problem. So as many of you will know, there is an existing ethics framework. It's pretty popular. The reason I'm showing it to you is that it's been applied in 2004 to vaccine trials in general, and again in 2016, and again in 2022 in the ERV VaxLit manual. So for those of you that are familiar with it, please bear with me, but I really just wanted to make a few brief remarks about it in relation to discovery medicine, and some people on the call may not be familiar with it. So this framework really argues that to be ethical, a study should have these various components. If you just start with the first component, it should explore a question of relevance to a significant health problem in the setting, and that's this ethical concept of social value. And then it should use methods and procedures that will yield reliable and valid results and findings, and that's the ethical concept of scientific validity. So some people would challenge, why is that component part of an ethics framework? And the answer generally is that without component one, participants will assume potential risks, burdens, inconveniences for no benefit to society in the form of societal knowledge gains. And obviously, we've heard from the first speaker that in these trials, the focus really might be on trying to generate scientific insights. So, for example, how to choose immunogens and regimens that could eventually design products with a better chance of success in the field. The second component is that for studies to be ethical, participants need to be selected fairly, and that has three parts generally, as some of you will know. You obviously researchers must select for science, not for vulnerability or convenience. But then of the scientifically eligible, they need to select to minimize risks to participants. And then finally, to ensure that there's a fair spread of risks across all the collaborating parties. And of course, for these trials, the participants that will be selected are likely to be at low risk of HIV infection, and that would be appropriate. It would be fair, given that the purpose is to achieve scientific insights about the immune system and other important scientific questions. The third one is that the potential risks of the study procedures and interventions to participants must be sufficiently mitigated, and then obviously sufficiently outweighed. This can sometimes be by direct potential benefits to participants, or by societal knowledge gains. And this is that concept of a favorable risk-benefit ratio. And of course, for these particular studies, it is clear that what is the compensating reason that outweighs the risks to participant is the societal knowledge gains. And as the first speaker mentioned, it's very unlikely that there would be a potential individual benefit to the enrolled participants themselves. And if you look at the fourth one, it's that there should be ongoing respect for participants after they're enrolled, and this can include a whole basket of issues, including a standard of prevention, access to care, fair payment, and access to results. And this is sometimes the concept here is the ethical concept is ongoing respect. And of course, applied to these studies, participants should have access to prevention methods, even though they may be at low risk of acquiring HIV. There should be some careful plans for how to ensure care for them, even if it's highly unlikely that these participants may acquire HIV infection. There should be a fair payment to them that's commensurate with the time spent at sites and some of the inconveniences of the study procedures. And very importantly, in these trials where there's not going to be a product, the benefit here is access to results. In other words, access to the knowledge, what are going to be the scientific discoveries, and making sure those are disseminated to all the affected parties. And then number 5, ensuring that stakeholders are appropriately engaged. And what this can generally mean, if you look across guidelines, what do guidelines value about stakeholder engagement? What do they think excellent stakeholder engagement is? It's usually that it's broad and diverse, and that it's early and sustained, and that it's responsive to the context and dynamic over time, and also that it's well described in a transparent way and adequately funded. So again, it would mean that in order to have good stakeholder engagement for these studies, we would need to make sure that those persons that are affected are engaged in a meaningful and non-tokenistic way. And then number 6, ensuring voluntary informed consent from participants. Applied to these studies, it will be essential, as the first speaker has already described, that participants, one of the real central concepts, almost a deal breaker, is that there's no possibility that the intervention will confer direct individual benefit to themselves, but really the value is in the societal knowledge gains. And lastly, that the study has to be reviewed by a competent independent research ethics committee. Jim, next slide, please. So the article that was shown earlier the Prudden article has already identified some of the issues that you can recognize as ethical issues. And here's just a selection of them. So they argue that CABs and community stakeholders will need to appreciate the central purpose of these trials, and that might require very, very sensitive, tailored explanations. That the risk benefits of the trials will need to be carefully communicated, and that the purpose of the trials in consent materials and in consent forms for participants they need to understand that that there shouldn't be a misunderstanding that the study intervention will confer a direct health-related benefit to them, or even that the study goal is to move products through a pipeline towards introduction. The authors also made recommendations that study teams really need to do some careful work to elicit the perspectives and opinions of community stakeholders and advocates who will need to be engaged, and that countries may be involved in these programs that may not have been accustomed to it being engaged in early phase trials before, and that RECs may need to network with each other to harmonize any concerns and perhaps their recommendations. Next slide, please. It is likely, sorry, if you could go one back, Jim. It's likely that it could be helpful to have a much more systematic exploration of what concerns are. So our group is planning some empirical research through the South African Medical Research Council. We're really aiming to explore what are the perceived complexities and opportunities in these studies by engaging in in-depth interviews with a range of experts all the way through from RECs and bioethics experts right through to international advocates. What we're really chasing here is we're trying to understand the degree to which our current ethical guidelines will address or accommodate or resonate and respond to the perceived complexities, or whether in fact there are gaps in guidance. Next slide, please. We've started with a review of guidance, and given that the benefit, the post-trial benefits in studies like this is largely access to knowledge, we've started a review of guidelines to see really what do they recommend in terms of who should receive access to study findings, when that should happen, to see whether these recommendations are helpful, as well as a whole range of other issues we're exploring. Next slide, please. And finally, we're striving to develop a resource largely for advocates that highlights ethics recommendations and how they might be a helpful source of influence to advocates if they're used strategically. And here we've had inputs from CASPA partners and collaborators, including from the HVTN, one HVTN representative, given that they've had a lot of experience with these trials. Last slide, please, Jim. So to close, we think that this shift to discovery medicine for HIV vaccines is an opportunity for a deep dive into current ethical guidelines, really to assess their adequacy. It's an opportunity to raise awareness of existing ethical guidelines and an opportunity to anchor advocacy practices in existing ethical guidelines where this is going to be helpful. That's the end of the talk. Thank you so much for your attention, and back to you, Stacey.
Thank you, Cathy. That was such a great overview of those issues, and look forward to discussing further, of course. And I'm going to quickly shift to our next set of presenters. We've got two really great presenters who are going to talk to us about a somewhat new project that's really looking at this discovery medicine for HIV vaccines called the Brilliant Project. So we have Dr. Betty Mwesigwa, who is the Deputy Executive Director of the Makerere University Walter Reed Project in Kampala, Uganda, and is one of the PIs on the Brilliant Project, as well as Tian Johnson, who is the co-principal investigator on Brilliant for community engagement and advocacy. And one of the reasons we really wanted to bring this set of speakers on is because it's focused on discovery medicine, but also really a core component of Brilliant is their community engagement component. So very excited to hear from both of you. So I will pass over to you, Betty.
Thank you, Stacey, and thanks to the previous speakers for such a brilliant job. I feel like I've heard many of these things, but I feel like I don't hear enough of them. So thank you for leading us into the next presentation, and Jim will be presenting our slides. So my name is Betty Mwesigwa, and been already introduced, and very excited to be speaking today on behalf of the Brilliant Consortium. And we're hoping to do brilliant work as we take on this discovery medicine journey for an HIV vaccine. And as the slides come on, I can just speak about Brilliant. Thank you, Jim. Okay, we can go to the next slide. So the Brilliant Consortium is funded by the US aid, and it was set up to harness and catalyze African scientists to contribute to an African-led effective HIV vaccine. Sandhya has done a very wonderful background introduction to everything we want to say today. So there's been lots of work going on in the background, but we still don't have a vaccine. And much of the work has been started and led and designed from the high-income countries, obviously working very closely with scientists and sites in the low-income countries. But this time, Brilliant is up to ensure that African scientists can directly contribute to HIV vaccine development through design and development of an effective HIV vaccine. So Brilliant is African-led, and hopefully that we will be able to develop some novel ideas, discover some molecules or products that could potentially work as HIV vaccines or be developed into if they're found to meet purpose. And therefore, everything we're doing is really discovery medicine at this point in time. We don't have any idea how far we shall go into clinical testing, but the hypothesis that Sandhya has, we need to generate new innovative hypotheses, either using already existing scientific information or doing all the discovery work to discover new ways in which potential molecules could be developed. Next slide, please. And so Brilliant is a consortium predominantly led by African women scientists. And of course, as everyone knows, a multidisciplinary team, scientists, research, clinicians, community advocates, and, administrative personnel, collaborations, a lot of work going into creating this consortium. And our overall objective is to evaluate HIV vaccine candidates that are going to be designed, developed on the African continent, led by African scientists. But as we do that, we will also be looking to test some concepts that are already existing, but would be first in Africa. For example, there are immunogens that might be already existing, but have never been tested in Africa. And obviously, we know how different populations can have different immune responses to different products. So we do need to do some testing here as well. So we will be doing some first in Africa concept testing. But the whole point is to rapidly evaluate African immunogens by doing discovery, preclinical evaluations, including different platforms, not just the mRNA platform technology that we quickly got to learn about and love from the COVID experiences. But we could use all other concepts that are already known to be able to quickly accelerate HIV vaccine development. Next slide, please. Oh, sorry, I hope you have all seen from the map, there's several countries involved, but these countries in Africa will be working closely with other developers in the Western world. But Brilliant is made up of all the countries on the slide, but led by the South African Medical Research Council. Discovery, I don't even want to get into because I think Sandy has done a great job, but it is finding, getting out to do a search, and then you find or bring to light new things. And discovery medicine is here to help us identify molecules that could potentially be translated into products that can be used as HIV vaccines. So we're at the discovery phase, not necessarily at the linear HIV vaccine testing phase of phase 1, 2, 3 to find a product. But ultimately, whatever products we look at need to be safe and need to give us hope for inducing immune responses that could be protective against HIV. So for Brilliant, we're set up in several programs. We have a community program which is led by intending to partner with civil society and to advance the community components of vaccine discovery. We have a clinical program which is responsible for designing the studies and ensuring that they're well implemented, the sites are ready, the sites are implementing well to try and ensure that the molecules are safe, but also give us an immune response. Like Sandy mentioned, sometimes we might have to go back and forth to look at the products again to keep improving the immunogenicity or the immune responses that we are getting from any of the products we'll be testing. And therefore, the clinical program works very, very closely with the laboratory program, because as we look as we give these products to humans, we do need to test to see exactly how the body reacts to them. But we also have exciting for African scientists, the discovery and preclinical program, where new innovative products, and Sandy has already mentioned, for example, the MFV concept. If we took that on and looked at the clades that are circulating on the African continent and developed something novel, but following that already known science, and then test it in animals before we get into humans. So it is going to be very exciting, especially for African scientists to participate in the discovery and preclinical program. And then obviously, as we do all this, we're being very intentional about collaborating with other scientists in the world, because we don't want to lose any, we don't want to reinvent the wheel. but also ensure that there's enough capacity on the African continent to be able to do a lot of the complicated testing and discovery that has been done elsewhere. And hopefully together, we can come up with an effective HIV vaccine. The rationale for which is clear to everybody on this call, I guess. Otherwise, we wouldn't have joined the call. Next slide, please. So what is the scientific strategy that Brilliant is taking? So overall, we will try and test immunogens that have been tested in humans elsewhere. So we're working with several developers and looking at the products that they have developed and have tested elsewhere, but have never tested in Africa. So that we bring those and do first in Africa studies just to quickly advance the science. You know, if they have done it in one way, we being innovative and doing it in different ways so that we can learn faster about those products and what they could do. And also, we will want eventually, so using already developed products helps us to test the consortium systems. There's many countries, different regulations everywhere. So we'll test the system with existing products, but in the meantime, advance science by trying to develop our own products. Next slide. So I just put this slide in to give an example. We're already in the process of creating the first protocol using these products. These products are already being tested elsewhere. So we thank all the collaborators that have given us these products. Roger Sanders, Carrie Caspers, and then Leo from different organizations going to work with us for us to develop protocols that can test these products in novel ways, different from what they have tested, but different enough to maintain harmonization in the way we can interpret the data. So this, for example, will be our first trial, which is expected to start at the end of this year, and to help us, kickstart the system and get all the different sites ready to do the discovery medicine that we're excited about. Next slide, please. And then as that happens, we will develop immunogens that are purely developed by brilliant scientists, but also increase capacity to develop them into vaccines using different platforms like mRNA platform. We're also looking to collaborate with the MHRP on the MFV concept that Sandhya has already described. We also have a collaboration with the University of Amsterdam, where we're going to have scientists trained on development of SOCIP trimers, but also we will be looking at other epitopes that could be useful for generating immune responses and development of vaccines, HIV epitopes. So it is exciting times. Let's continue, please. So this just shows that the Amsterdam team will be working closely with the Joint Clinical Research Center here in Uganda. Joint Clinical Research Center is a research organization, but part of the Brilliant Consortium. And there'll be training scientists, like I mentioned, at Roger Sanders' lab. And then when these scientists come back after their training, we're hoping to have tech transfer on infrastructure and systems and expertise for us in Uganda to be able to actually develop SOCIP trimers under Brilliant. And hopefully in the end, we'll be able to diversify a pipeline for testing of products. Next slide, please. And I'm delighted to be leading the team here at Makerere University Walter Reed Project, working closely with Sandy and Morgan at the MHRP and other members of that collaboration to develop or to move forward the MFV concept. But this time we want to be focusing on subtypes that are circulating on the African continent because the MHRP team are focusing on the subtype B, HIV subtype B. But like we said, we don't want to be duplicating, we want to be innovative. So we will try and look at what is circulating in the African continent. This work is yet to start, but conversations are ongoing and very exciting. So watch this space. Next slide, please. And we also don't want to be doing just the mRNA. We will look at other platforms, for example, proteins, and try and ensure if one platform doesn't work, we still have hope with others. Next slide, please. And also, as all this happens, we will be getting a lot of tech transfer in the assays and testing lab assays that we'll need to use for the new products, but also for the existing products that we'll be testing that have not been done on the African continent or that is not available on the African continent. Even then, if one of the African countries in the Brilliant Consortium has technology that could be transferred or to another lab, we're looking into both local continental transfers, but also across continent transfers of different tech transfers for even the testing assays, lab assays. Next slide, please. And that's it from us. Hopefully we'll have more chances to discuss the work that Brilliant will be doing for discovering an HIV vaccine. It's been amazing speaking to you. So I'll hand over to Tian. Thank you.
Thank you, Dr. Mwesigwa. I really appreciate your presentation. Lots of great information there. And so at this time, I just want to open it up for Q&A and discussion. Just a reminder that we have lots and lots of links for relevant information about all of the things that we've talked about during the presentations in the chat. So if you're looking for any information on some of the things that have been presented, please look for the links in the chat. And you still have opportunity to go ahead and submit questions for our Q&A discussion. So if you have any questions for any of our speakers, please submit those in the Q&A section right now. And so,
Hey, Lewis, this is Jim. I think, was Tian going to speak? Tian, are you speaking to these slides here?
I am indeed, Jim, whenever you're ready. Hello, everyone.
Okay, sorry for any confusion. Tian, I think we're on your first slide here. Just let me know when you want to advance.
sorry, y'all, off the gun. Go ahead.
No worries. Could you go back a slide, Jim?
You bet. Hold on.
Thank you. Hello, everybody. Thank you to AVAC and the Choice Agenda for the very kind invite to spend some time with you today as we reflect on where we are in the long but essential path to an HIV vaccine. It's such a pleasure to be here and see you all. My name is Tian Johnson. I'm the founder and strategist at the African Alliance. We're a Pan-African queer-led health justice organization that works to advance accountable and radical, authentic leadership in the global health landscape that secures Africa's voice and preeminence in this space. I'm the co-principal investigator for community of the Brilliant Consortium, a member of the Pandemic Funds Advisory Panel, and the International Civil Society Observer of the Robert Carr Fund. I'm presenting here today alongside my two co-leads of the Community Engagement and Advocacy Workstream, Martha Tolana from GALS in Zimbabwe, and Stefan Mugamba from Makerere University, Walter Reed Project, both critical and long-time leaders in the community engagement and advocacy space in the context of clinical research. I want to acknowledge the leadership of Professor Glenda Gray, the principal investigator, and Linda Gale Becker, who serves as co-principal investigator of Brilliant. I want to make special mention of our other two co-principal investigators, Black African women who have and will continue to lead us, who have and continue to show us the way, and who have and continue to keep this work honest. Dr. Sisi Kitio, Dr. Betty, who you've just heard from, we name you, we see you, we honor you, we celebrate you. We recognize your dedication, your commitment, and your leadership that sets the tone and raises the bar for what governance and leadership of an HIV vaccine research must be. Congratulations to Dr. Sisi today as well for being named as one of the highest ranking scientists in the field of microbiology in Uganda. Colleagues, I want to speak a bit about the history and the context and where we come from. We've always worked for clinical research that is accountable. We've always used a lens of decolonization. We've always asked what it means in terms of power, how power is wielded, and how power is expressed within clinical research. We've spoken and advocated about representation around voice and visibility in the HIV vaccine research space. We have a long history of partnership with the South African Medical Research Council, who serves as the prime lead of this truly African-led work. Part of the reason why we were attracted to the work of this consortium was the unique nature of the opportunity that gave criteria around leadership and decision-making that we had not seen previously. There was a clearly articulated request for meaningful leadership positions across all decision-making and implementing structures of this consortium, of communities, of women, of a diversity of communities. The power of seeing a funding opportunity that had the courage to name us, to acknowledge us, to see us, made it clear that this partnership was an opportunity to advance a new level of consciousness in the HIV vaccine research field. We knew that this was a space where our identities would not be diluted, where our advocacy would not be managed by those who were not African, an opportunity that walked the talk of inclusivity in measurable and accountable ways. It's a testament to the USAID team for practicing humility and a genuine interest in what our needs and priorities as community within Brilliant wanted to see, wanted to do, wanted to create, not just within the consortium, but as contributors to the field as a whole. Our realities are valid, our stories are our data, and our history is full of examples of the benefits and simple logic of meaningful community engagement in research. The work of this consortium, which I'm going to speak to in a bit, also allows us an opportunity to secure skills building of advocates in ways that we have not seen before, by curating along with leading global institutes using principles that work for us as Africans, an adult learning experience based on immunology and vaccinology. So while this will not turn us as advocates into scientists, it will give us the language we need to sit around that table and engage meaningfully, engage with authority. And that is what attracted us to this consortium that we're really, really excited to be a part of. So now I'll get into some of the nitty-gritties in terms of the work of the community engagement and advocacy work stream. Next slide, please. Really, there were 4 areas that we, as community activists and representatives, bring to the Brilliant table. It's such a spectacular privilege to work with these amazing scientists and researchers and thinkers. But we really wanted to hone our focus on four key pieces. The first was around community governance, community leadership within the scientific consortium. The second was co-creating an advocacy agenda around discovery and what that means. We regard this as a new and exciting challenge as part of number 3, where we look at communications. We have not really done a lot of work in terms of communicating these concepts around discovery medicine. It was really, really important that we stood up and said, let this be the time where we bring communities along for this discussion and this absolutely critical work. And lastly, and perhaps even more importantly, was the positioning of the monitoring and evaluation component of this entire consortium within community. We're really, really fortunate as the community engagement and advocacy workstream to be the home to the monitoring, evaluation, and learning component of the entire consortium. And I'll speak more about that in a minute. Next slide, please. So, as I spoke about good governance, we walk the talk. Community is embedded in almost every single structure of the Brilliant Consortium that makes decisions, that shapes and guides the work of the consortiums. Leadership from the scientific advisory group, which is the highest decision-making body of the consortium, right all the way across to subcommittees working on clinics, on labs, and of course on community engagement, on MAL, etc., etc. So we're really excited about what the potential is presented to us around having communities so embedded in all decision-making structures across the consortium. Next slide, please. The second piece, of course, was rolling out this work. We want to walk hand in hand with communities across these 8 countries. We want to contribute to a growing field of knowledge around community engagement and raising our level of awareness and understanding of basic scientific principles, of research principles, of discovery principles, and what it means. We've already started working on a 40-piece compendium that explains basic discovery principles to communities in a range of contexts and languages. We've worked with partners to ensure that we hit the ground running and curate a selection of materials with and for communities that will allow us to begin to continue the discussion. Because of course, we don't view this as new work, we view this as building on the historical work, the historical investments, the work of partners like Casper, Advanced, Matrix, to move the field ahead in a cohesive way, but whilst maintaining that momentum that we absolutely need to continue this work. Next slide, please. The second piece, of course, was developing an advocacy agenda around Brilliant. But of course, what that means is we take our partners, communities along with us, and we co-create this advocacy agenda at country level, but also at regional level, that can begin to articulate where communities see themselves apart from in these decision-making structures, but more hyper-localized decision-making structures at country level, at regional level. The African Union, the Africa CDC, SADC, ECOWAS, etc., is certainly not out of our scope, and we will be making conscious efforts to engage and make the circle bigger in terms of opening up a space that we regard to be exclusive and has been exclusive for a while. So this is an absolutely exciting opportunity for us to begin to break down those barriers that have stood in the way of community and science for too long. Some practical ways we've done this, of course, is incorporated HIV vaccine discovery modules into existing community toolkits that will be rolled out again in a range of languages. All of the work that we do at the African Alliance is really grounded within this understanding of decolonization and what it truly means to center our responses in African realities. And to that end, of course, we honor our traditions and our history of storytelling, our amazing legacy of being able to tell a story in a compelling way. And in this case, HIV vaccine research is no different. So we're really, really excited at looking at a range of modalities around how we begin and continue to communicate, be that community radio drama series, be that fireplace stories, be that hands out under a tree, etc., etc. So we really want to embrace the complexity and the depth of what true, authentic community engagement looks like on this continent. Next slide, please. As part of this work, of course, and walking the talk of decolonization, we don't implement in countries. We recognize organizations, systems, movements, networks that have existed. And it's really, really important for us to walk that talk and engage with existing organizations who are our partners across the historical microbicide field, the condom field, the PrEP field. The landscape is ripe. The setting is there. The goodwill is there. The trust is there. The passion is there. And there's no reason why that cannot be brought over into the work of Brilliant. And so we absolutely want to take advantage of the spectacular investments that have been made historically to get us to this point where we are today. Next slide, please. So a principle I've spoken about, engagement with SADC and the regional bodies to ensure that political principles, those who claim the title of leader, are aware of the work that we are doing and have a working knowledge of the efforts that are being made from Africa to advance the field. Next slide, please. Of course, communications for meaningful engagement is one of our biggest focus areas. It's a focus area that really excites us, and we're really keen to get going with a lot of the pieces here. As you can see, we've curated a selection of modalities, of tools that communities can use, ranging from newsletters in Shona in Portuguese, in Pidgin, in Nyanja, in Swahili, to podcasts that look at both the scientific leadership at country level and have a discussion around accountability of science at a country level in the context of the consortium, but also profiling community advisory board leadership. Also profiling activists who are perhaps not part of a CAB, but who play an equally important and powerful role in holding science to account and ensuring that scientists and researchers not only know that we are watching, but we are around the table and we are with them. Because at the core of this, it's an exercise in trust. But we want to move from trust into true accountability, curating a series of consortium-wide op-eds that capture the moment, that capture the nuance of the work that we are doing as a collective, but never losing sight of the power of the national story, never losing sight of the power of these country examples, country leaderships, countries that are standing by scientists, investing in research, countries that have created an environment for the work of Brilliant to happen in the way it is going to happen. Journal articles, publications, presentations, profiling all of the consortium sites. Our work is really cut out for us within these next 5 years of the consortium in terms of how we honor, how we center, how we celebrate partners who have been doing this work for decades now, but really positioning that celebration within the context of what it means for HIV vaccine discovery. Next slide, please. Utilizing existing participatory strategies, looking at MAL that I spoke about earlier, but our MAL really has given us an opportunity to meaningfully think about what accountability really looks like. Whose voices are we hearing in the field, in the consortium? Whose faces are we seeing in the field, in the consortium, scientific and community? But the work of the community engagement and advocacy MAL piece is really centered around understanding power, understanding how power manifests, how power is used to advance the work of the consortium like Brilliant. All of our work we look at through a lens of gender and race. It's absolutely important to us. The personal is political, and we never ever forget or dilute our roots, whether we're working in HIV vaccine discovery or condom programming. So within year one of the consortium's work, we will be able to share with you, hopefully on another Choice Agenda call, some of the initial findings in terms of how we've been able to track voice and visibility and representation, based on posters which we presented at conferences nearly a decade ago that were entitled, "What Communities Want Scientists and Researchers to Know." So none of this is new. Brilliant has presented to us as community activists and communities an opportunity to finally solidify and pin down some of these key concepts and thought processes that have really been on our mind for far too long. And so it's an opportunity to make that progress in the context of the work of Brilliant. Next slide, please. And so that is a flavor. It's early days. We hope to come back and, of course, share some of the practical examples and profiles of what we've done as the community engagement and advocacy leads of Brilliant. But, in closing, we're excited about what Brilliant represents and its potential as we work together to honor these investments that have been made, the memories of those that are no longer with us, that have gotten us here in terms of a quest for a vaccine. But the work is about more than a vaccine. It is about our dignity. It's about our prosperity as Africans. It's about the world we will leave behind. It's about proving to ourselves that when we come together with singular focus, determination, and perseverance, we can deliver an HIV vaccine in our lifetime. We don't have a choice but to make this work. We must ensure that we truly leave no one behind. It's hard and it's difficult work, but our work to find a vaccine must be fueled not only by the urgency of the challenge, but by the unwavering belief in the power of human ingenuity and our collective determination. With every trial setback, with every breakthrough, we draw closer to a world where HIV is a memory, and hope for our freedom, happiness, and ability to claim our birthright of living lives that are full during our time on this planet must always remain at the center. Thank you so much for having us, colleagues.
Thank you so much for that brilliant, brilliant speech and presentation, Tian. Yeah, those are powerful words, and it truly resonates with me. Our goal in this work should be to put ourselves out of work, and we should always remember that. And we're in the business of saving lives and changing lives. And so that's what an HIV vaccine is really all about. So thank you. So we have a little bit of time left, and I want to launch us into our Q&A section. So again, just referring you all to the chat if you are looking for more information about some of the things that have been presented there throughout today. Plenty of links there if you're looking for more information. We have quite a few questions already in the chat, and actually our speakers are actively answering questions in the chat. So just be mindful that if you look in the Q&A section, particularly, there are some questions that are answered. There's little tabs at the top of the Q&A section if you click on it. And if you look at the top, there's open questions and then there's answered questions. So you can see some of the answers, some of the responses to questions from our speakers today. But for the open questions, we have quite a few questions around more nuances around discovery medicine and asking for more examples and kind of strategies around things like community engagement, ethical considerations, and then also thinking about what are some of the key or prominent things happening in the discovery medicine space. So I just wanted to turn it over to our speakers today. Can you talk a little bit about or give some examples of some of the best community engagement strategies that you've heard in discovery medicine, and also maybe some of the best examples of ethical considerations that you've seen in discovery medicine? And then, for folks like Sandy, do you want to give some of the what are some of the hot topics or the bright spots that you're most excited about?
Louis, it's Cathy here. Sorry, for some reason I couldn't put up my hand. I'm happy to respond to the question about disproportionately affected communities. Should I go ahead? Right. So the question, thank you. I mean, it's a very good question, and I think what we must bear in mind is that disproportionately affected communities are the intended beneficiaries of an eventual vaccine. But the reality is that they may have simultaneous vulnerability factors. So it's critical to spell out exactly what those might be and to get quite granular about it. So for example, participants may have low educational attainment. They may have low research literacy. They may reside in impoverished settings. Their community stakeholders may have power differentials relevant to the study team, and then consider what are the appropriately dosed remedies. And that requires a whole lot of strategies. I'm not going to talk much about the community ones because Tian gave an excellent overview of what a comprehensive community strategy looks like. But for example, you would need to have ways to strengthen the informed consent of participants. You would need to ensure that offers made to participants like payment, are very well justified and approved by research ethics committees. You want to empower community stakeholders so they can impact the agenda. The whole idea would be you want to make sure that participants and community stakeholders drawn from disproportionately affected communities can be involved in such trials, and not neglected for involvement, but involved in a way that respects their rights and welfare. Back to you, Louis.
Thank you so much, Dr. Slack. Sandy, do you want to talk about any prominent studies or prominent interventions in the discovery medicine space? Anything that's got you truly excited right now?
Sure, Louis, but I actually start by answering a question you asked earlier, which I think may resonate with this group. You asked about effective community strategies. And one of the things that has struck me, I serve in a different forum as a co-PI of the I4C Immunotherapy for Cure Martin Delaney Collaboratory, which AVAC is involved with directly as our community liaison. And we're bringing the voice of the community further back into the basic science and preclinical space, because so much of community engagement is appropriately focused on human work, and human clinical trials, which is the subject of this webinar, but having more of that voice, even in the earlier stages of development and discovery, and hearing feedback on this may or may not work, I think is equally important. And we're just starting to get into that. And I'd love to see more of that, especially as there's more and more basic discovery moving directly onto the continent of Africa and other LMIC. I think having the community get more involved in the scientific upstream work is going to be equally critical. And so that said, for your second question, just briefly, it's hard to kind of qualify all of the many approaches that are exciting. I saw that Roger Tatou mentioned adjuvants. So, I think for vaccines, just to kind of put the themes very broadly, there's a focus on additional compounds called adjuvants to improve or broaden the immune response. There's a focus on combining different types of vaccines. Of course, mRNA vaccines are newer. Could they be manufactured locally in host nations so there's immediate access, and then combined with other regimens? And then a focus on the types of delivery, maybe not, where it's injected or with what schedule or changing dosing. That's kind of a very superficial summary.
Thank you so much. That was great.
Maybe just to build on that and just to acknowledge we are over time, which we somewhat expected, but just again to thank all the speakers. I mean, those were, not to overuse the word, but those were really brilliant presentations. And Tian, I think you ended us on such a powerful and important note. We will indulge everyone and take just a few more minutes to, I think, make sure we do have a little bit of discussion. But I guess maybe Sandy and for Betty to kind of, I think, build on this sort of issue around, all of these sort of scientific variables that go into developing a vaccine. You've got the adjuvant, you've got the platform, you've got the regimen. Most importantly, I think you've got the immunogen. And really interesting, I think, Sandy, to hear a little bit more kind of detail on what's exciting and what's really moving forward. But we've heard kind of bits and pieces of different projects that are happening. I'm curious if you could talk about how are different research groups kind of sharing information, and kind of making field-wide decisions about progress and where we're seeing success and how we're really kind of bringing all of these variables together for an effective vaccine.
I was watching Sandy very closely. So thank you. So how are scientists sharing information? So I think we have all understood that there's no way one group can work alone. Multidisciplinary is critical. I think the other critical thing for us to remember is that the work has been going on for nearly 40 years. Trying to find an HIV vaccine is not new. So all the groups are learning from each other and trying to advance the science. And the only way is to collaborate, to speak, to ensure we all hold hands together because of this one common important goal. So I'll tell you, from working with MHRP for a long time, I know that you might find, for example, the 591 trial that Sandy has just described is happening here at our site. The different parts of the regimen, the different products are coming from different developers. So the sponsors, the funders are different. So people have a different hand in trying to advance this one trial. And I think that's what's happening a lot with all the scientists. For Brilliant, I can't tell you how many conversations we're having with how many people, because we understand how important what has been learned before is, as to try and help us, think what else could be done to advance the science. So there's a lot of talking across consortia. There's a lot of discussions with, while Tian is discussing brilliant, he's working very closely with Casper to ensure that we're not confusing the communities, because they have had so many messages over the years. So Stacey, collaboration is critical. Multidisciplinary teams are critical. And of course, meeting and attending scientific meetings, tech transfers to ensure people, sharing new things as they come through is all important. That's what I can say. But Sandy, maybe you want to add anything?
I think that was very well said. Maybe one important and really, it's worth mentioning, the more that we come together, unfortunately, we're seeing this from COVID, the more that these legal agreements between parties becomes a factor. We would really love to just see the world come together and work for the greater good. But when we bring in different groups that have different products and different intellectual property, that's becoming an increasing part of the upstream dialogue to make sure that all the players can work together in concert. And I've come to acknowledge that you have to really tackle that piece early in order to develop a successful joint collaboration.
Great. Yeah, I think collaboration is oftentimes easier said than done, but really great to hear some of those sort of concrete ways it's being looked at, especially in the sort of discovery medicine stage. We do need to wrap it up, and I'm wondering if any of you, maybe just one of you, would like to chime in and just say. I mean, I think a lot of this sort of Q&A has been about how we're ensuring that this sort of vaccine discovery is really kind of moving forward in a successful way. And I think, Tian, I'd love to hear from you, because you gave such a great sort of overview of how this work really builds on decades now of sort of community engagement, and I think many stakeholders kind of ushering forward this search for a vaccine. And curious if you could kind of say, what's your hope for success, a year from now, in your role in Brilliant?
Yeah, thanks, Stacey. I mean, I think our hope is also really regulated, because if there's one thing this field teaches you, is humility, and to wind things back quite rapidly in very big ways, and really think about how much we're deferring this dream. But I do think, after a year, I'd love to see communities at the very least in the 8 countries where Brilliant works, with their needs having been met. We always say that HIV vaccine research doesn't happen in a vacuum. There's a context to this. The context has not always been pretty. The context has not always been on our side. The context has not always acknowledged our diversity and our complexity as human beings. And we've seen science fail so many times when it has not done the right thing by meaningfully engaging communities. And so I really think this first year of Brilliant presents to us an opportunity to lay further groundwork, further foundations on not only acknowledging the historical work, but really practicing humility in science, humility in engagement, which is a very, very tall order within this current field. But it's something we believe is absolutely worth the fight. It is worth our time to keep scientists' feet to the fire. It is worth our time for researchers not to sleep without knowing that we are watching them, we are holding them account, because their work is not for the next Lancet piece. Their work is not for the Nobel. Their work is for us. It is for communities. And it is because of the work of Black African women, scientists and researchers, that the world will have an HIV vaccine. And that's what gives me hope and drives me. Thanks, Stacey.
I love it. So leaving off with humility and holding researchers' feet to the fire. Couldn't have chosen better words myself. Thank you so much. And thank you so much to all of our speakers. This has been a great time, a great moment for us to get together and celebrate moving forward towards an effective and safe HIV vaccine. So I commend you all for your work. And I encourage you all as you move forward. This work is sometimes a heavy lift, because sometimes we don't see the light at the end of the tunnel. But as has been said in multiple ways throughout the day, you know, we're in the business of saving lives and changing lives, and we need to always remember that. And so thank you all for your time, your passion, your energy, and your expertise. Thank you to everybody who's joined us and stayed on a little bit longer today. We really appreciate you in this space. I want to say a personal thank you to the Choice Agenda and AVAC for hosting this and allowing me to be a part of the space. I really do appreciate it. Just remembering that we will have a document that summarizes all of the links and information that was put out in the chat. So if you're looking for any of the information that came through the chat, that will be put out later on. And this recording will also be made available publicly. And so with that, just thank you to you all. I hope that you all have a wonderful day, a wonderful evening, and, you know, let's push forward in doing amazing work together.
My feet are burning,
Thank you
Tian.
so much. Thank you. Thank you.
Bye.
Thanks all.
Bye, everyone. Thanks so much.
Thanks for
Bye.
setting that fire, Tian.
I know.
Amazing
We're all
work,
feeling
Tian.
As ever. And thanks everyone for being here. In the next day or so, you'll get a follow-up email with a link to the recording, the slides, and that resource document that Lewis was talking about. So with that said, I'm going to close out this Zoom. Thanks all for being here, and I hope to see you all at our programs in July, and enjoy a month free from TCA webinars in June. Take care.
Thank you, Jim.